Hey guys, it's Alexander Williamson here with the secret history living inside your aquarium. Today we're going to be talking about plant tissue cultures, and we're going to talk a little bit about the history having to do with them. Now, it is really hot in here, so bear with me. I'm upstairs in our house, which gets scorching. So it's a live stream. Feel free to interject with questions, as always, and the more the merrier in here. And this episode today is brought to you by Dr. Pepper. They're not paying me, but they should be. What's up, Bentley? Welcome. So, I'm going to be talking about the history of plant cloning and the history of and the science of tissue cultured plants. Cloning plants, same thing. As well as maybe showing you guys how to do that. But more so, I just want the first part of this. I'm just going to give you guys some raw history information because it is the secret history living inside your aquarium. And I feel like I've been slacking on those videos lately. Welcome, Soggy Pits. Come on in. So let's get right to this. If you have questions, line them up and I'll shoot them down after I give my little spiel here. The spiel is kind of so that people can watch later and get this as basically it's kind of like a live no retakes, no outtakes, one time deal of tissue cultured plants, the history of, and how cool it is that we have them and that you can actually do it yourself in theory if you know what you're doing. And it's not like rocket science. It's like cloning science, it's not that hard. Like you can clone people, whatever. So let's get right into it. So right here, you'll see I've got stuff lined up. You may have seen these in the hobby before, especially if you're into aquascaping. These are called tissue cultured plants, and these ones happen to be from ADA, the actual amono, aqua design amono over in Japan. But and actually, I think Singapore and Taiwan distribute as well. But you get these plants, and what they are is it's agar in the bottom, which is sometimes clear, sometimes dark. It depends on what the plant's roots look like and which type of plant you're getting. But basically, it's a gelatiny feeling substance, kind of looks like jello or something like that. Hello, Big Tex Tanks. Welcome. Come on in. We're going to be talking about tissue plants here, and then I will chat with everyone. If you have any questions about this kind of stuff, we're going to talk about who invented it, when they invented it, why they invented it, some of the weird connections, and then we're also going to talk about how you prepare these once you've bought a tissue culture like this, or how you can prepare your own. So I want to kind of go over a little bit of all that, and then questions we'll do towards the second part of it. And I'm hoping to get this covered in like 15 or 20 minutes, this part of it. So let's jump right in. Like I was saying, we'll continue right along. So in here we have a rare strain. It's like a pink flamingo, ultra pink flamingo. This is from Aquariums In in Seattle, but it was imported from Japan. And this is a very, very cool looking crypt. And normally you see a crypt when you buy it, cryptocorin, and you would see it as one little tuft. I can go show you real quick in the tank, the difference between things like this. So I'm going to show you guys. Welcome. Come on in, everybody. So, for instance, this is Mermaid Weed. It's also sawtooth plant. There is its Latin name right there for you. But this is a type of basically mermaid weed, and it looks like this normally. So if you go to the aquarium store, usually you end up buying plants that kind of look like a plant, and those are grown above ground, and they then have to turn into underwater plants. Cryptocoryns look like this. So a lot of times they're much denser than that. In my other tanks, they're much denser than that. But this is a newer tank, and I used all of the plants in this tank are these are all, I think, I'm trying to make sure I'm not lying, except for the lily. These are all started from tissue cultures, basically. So these have all grown up in the last 5 to 6 weeks from cups of tissue cultures. Now, let's talk about first why you would use tissue cultures if you're new to this. Basically, you use them because there are they are more expensive, but they are clean of all pests, bacteria, fungi, viruses, snails, anything like that that might come into your tank and wreak havoc. So that's one reason we use these in the hobby. The other is that these don't grow quite like a normal plant. So if you have a normal plant that's been from seed or that's been cut, as in a graft, or as a you cut it and then it grows. So let me show you one more time in another tank. So this one here is a Bacopa that is red. So in this tank, do we have any Bacopa in this tank? I don't think we do. Let's go to the other tank. So we're going to the other tank. So we've got Bacopa. In here, you'll notice that there's stems, and this is a stem plant, but they're all kind of coming from a tangle of one root ball coming out of the gel. And it's so hot that the gel is liquefying right now in the house. It's, I don't know, 80 or 85 outside, and our house gets 10 degrees hotter than that. But, so, pardon me if I look sweaty and crazed in the video. But here we've got Bacopa, just a normal green one, and you can see how it kind of grows, and it can get scraggly depending on the conditions, and it can spread out between spots, especially if you're not using any CO2 gas injection into your tank, which this tank does not use CO2. That tank over there, the little tank, sure does. So this grows a little differently. So the first thing when you use these plants, you actually get much more material as long as you're willing to wait and you grow it and take care of it. Let's see if we can zoom back out. Sorry, this is always a challenge on this new system. Oop. Oh, there we go. So when you grow these in, when you inoculate a tissue culture, so you can actually see it flopping around right now. So as you can see, they take a cup like this, they sterilize this cup completely. This cup is sterilized completely, and they, in glass containers is where they would usually do it. And a lot of industries do this. For instance, the russet Burbank potato is almost all the same cloned potato. Same with bananas. Every banana is a clone right now. Every yellow banana you get at a supermarket in the US anyways. Things we know as plantains are closer to what most bananas look like. But there used to be a different banana strain, and there was actually a banana sickness that killed off all of those. And banana candy tastes like the old bananas did. And I'm trying to remember the name of what the strain was, but they had to change the strain because every single banana died around the entire world by this disease that they got. So there is a danger to using the same plant, like cloning it, because it has the resistances it has, and if you keep cloning it for generations, eventually something is gonna pick away at its armor, so to speak, its evolutionary armor, and something will kill it. And when that day comes, it can kill every one of them. So doing this agriculturally widespread, while it might give you a consistent, like if there's a rose, yes, the Gros Michel, fungus murdered it. Thank you, Bentley. Do you remember, Bentley, what the new ones we have are called? I'm trying to remember. But in any case, I digress. So we have banana candy that doesn't taste like modern bananas because the flavor was constructed in the 1940s and 50s by candy companies, and then they just didn't want to change it in the 60s. People liked that flavor, and it was much more creamy, supposedly. People who used to eat those bananas, when that's what Dole and Chiquita and all those places made. Cavendish, thank you so much. You guys rock. Current is Cavendish. So yes, and the Cavendish banana is what we are all accustomed to. If you are overseas, you may have something slightly different, but for the most part, there's cartels that control it. So even if it's organic or whatever, Dole, Del Monte, you have these bananas. Before, the bananas were more squat and they had a much creamier texture. So let's continue. That's kind of one of the pitfalls of cloning. One of the great things about it is that every cell in there gets a new start, and it has a new start with basically the same expiration date as old cells. But in plants, in in the early 1800s, in 1838, there was a German scientist, botanist, and naturalist named Matthias Schwelligen or Schleiwingen. Sorry, I'm not even going to fake a German accent. But he came up with botanical cell theory. And in this theory, he basically broke down a lot of different categories of plants. But the one that applies to this is there are two types of plants as far as cell reproduction goes, and that's either totipotency, which is as in total, and those form a structure called the callus. That's Callus, and then those are actually cells that are cloned. But what they are is they're random cells from the DNA, and they grow from certain starting cells. So, say you have a leaf cell, they can grow in plants, they can grow the entire body, whereas in animals and fungi and mold, oftentimes you need to have the DNA extracted, whereas the actual protoplasm and RNA, I believe, I may be wrong on this, so you can correct me if that is the case, is within every cell in the plant, just like a stem cell would be in humans, and you can then basically grow the entire plant. Now, they knew something about this because they know for the last 10,000 years, people have been cutting plants. And for probably the last 5 to 6,000 years, there's evidence of people grafting plants. So with stem plants in your aquarium, you can simply cut them wherever and then replant them. They'll grow roots and you're good to go. With the other plants in your aquarium, oftentimes, you can also, if they won't grow roots, you can then tie them to a plant that's similar. They do this with trees all the time, with apple trees and things like that. And they'll cut a splice, they'll cut an angle, like here, let me try to set this up one-handed. They will cut an angle in the tree. So pretend this was a solid trunk like this. They'll cut an angle off a branch, and they'll literally tie on a new branch from an apple that tastes good. And they'll put that raw tissue together and wrap it with a cloth or a bunch of different things. Fighting traffic on a hot day in Seattle. Take care, Bentley. I know that'll be a rough one. It's always rough in Seattle. Okay, so that's grafting when they tie it together. And all apples, every apple plant you have, every tree, when it drops an apple, the seeds are going to be different. So every seed recombines all the DNA within that apple, and every single apple will be different. So you don't get pink ladies or red delicious from red delicious apple trees. You can only get a red delicious apple tree that's genetically the same by cutting a branch off of it or a sprout off of it and grafting it onto another plant. That, or by the 20th century, they started experimenting with tissue culturing, which is cloning. And grafting was kind of a form of cloning, basically duct taping, or bandaging two life forms together. And the problem that they seem to have with larger, more complex or newer plants is that those cells that they're using, so they cut the branch off a 100-year-old apple tree that's doing great. Well, in another 100 years, that's a 200-year-old apple tree cellularly, even though it's a 100-year-old tree theoretically. So it doesn't live as long, or it enters, not that plants have puberty per se, but if it were a mammal or a human, if you cloned it, it would have its post puberty self without the advent of hormone therapy and epigenetics. And they're just now still learning about epigenetics. It's part of the second green revolution. And epigenetics are basically codes within your DNA that turn off and on. Think of them as stop signs or flags, like when you're crossing the street, and they can either be up or down. And so you're born with the same DNA when you're a baby as when you're 80, but you don't look the same, right? So what's happening? One, the cells are not being recreated when you need a new skin cell. They only can be duplicated X amount of times in a lifetime of a human or of a animal. Now, in plants, there are many plants that can live for thousands and thousands of years, and it's wear and tear that actually grinds that down. But there's up to a 12,000-year-old tree. If you look at the quaking oaks, um, and quaking, what are they called? Quaking ash, I want to say. They are a tree that is all interconnected through the root system, and they have been alive for 28,000 years. Aspen, thank you so much. It's hard to do this from the top of your head, but I really wanted to talk about some history today, and I was getting jazzed about putting stuff in the tank. So here we go. There might be slight mistakes like that. And these are kind of things I'm ad hoc adding in to just illustrate points, because I find it interesting. And whether it's an aspen or an oak, if you looked up 28,000-year-old tree, you'd find that it was the aspen, and I had misspoken or didn't know. So yeah, so they can live a long time if the conditions are right. So with plants like this in an aquarium where you're really only hoping for 10 years or something like that, it really doesn't matter. And so you can go right ahead and do that. The soil is going to give out, or your tank will give out, or you will give out before the plants do at a cellular age. Now, when you start letting a plant grow 100 years and then taking it again, that's where we run into problems, and that's where the epigenetics are coming in now, and they're actually learning how to fix that. So epigenetics, like I said, are little flags in your genome, in your genes. So you've got your same DNA as when you're a kid as when you're an adult, and basically there are little switches that say you can either express this gene or not, and you can express these traits or not. And if you have, say, going through puberty one point in your life, where those little tags or flags are set off, like we're going to increase hormone production, and we're going to increase oil production in your skin and give you acne, yada yada. We're going to cause hair to grow. Well, plants also have these tags. All living things, even bacteria, have these tags. And now we're finding ways of turning them off and on and figuring out which gene is responsible. So it's not just the presence of a gene, as I talked about in the erythromicron danios that I just picked up last night. They have the epigenetic code that actually reverted. So they went back in time when they got stuck in the lake that they're in, and they took the form of their last known different species ancestor, and they basically stayed babies. So they told there's something in the environment or evolution, one of them or two of them actually remained babies and an inch long instead of 4 foot long carp or whatever they were. And they said, hey, this works for this shallow lake. And, that mutation was passed on or happened to be dominant, one or the other. But that is, you can watch that video from yesterday if you want. But plants have similar things going on where you can turn off and on these codes. And it was in the 1960s and 50s when, first of all, an Austrian man named Gottlieb Haberland, and that's a DT at the end. A German, Austrian, they were unified at this point because of World War II under Nazi funding, I don't know if he was per se Nazi, so I don't want to throw him under the bus like that if he wasn't. But the Nazis actually had really intense programs looking into growing crops more efficiently, looking into growing people the way they wanted. It can be used for evil or good. You know, there's golden rice that was genetically modified, and they put more vitamin, I believe it's A and B and iron into golden rice in the 1990s, and that ended up helping people in famished areas of Africa, in drought areas, and we basically invented this rice that could grow in those areas and that had the nutrients that rice normally doesn't have by splicing genes. So you can do some funky stuff with plants, and you can skip years of selective breeding and line breeding of plants, just as you would selecting traits in other organisms. And you can mix and match those traits. If you have a lot of money, you can do it right in the cells themselves. Whereas if you are like most businesses and most morphs, so you've seen normal green Bacopa probably, this is saffron red Bacopa. And that was probably an accident out of, there's 10,000 Bacopa plants in people's aquariums, and somebody noticed, whoa, this one had a defect, just like, a chromosomal defect maybe, or it could, and sometimes they're so bad that the plant dies, but other times, it's a benign one and doesn't matter. And it looks different, still grows the same. Well, instead of having to try to find another matching weird red one that's a 1 in a 10 million chance, now all you have to do is take a couple cells, and ideally you want to take the cells from the edge of a leaf right where the center line is, so where the stem kind of veins and stem are. And so you get some of that vein tissue plus the leaf tissue, and from that or the root tissue, you can take as few as maybe 10 cells, one in theory, but they usually use several thousand in a little sample. A sample that can be as small as, a cubic millimeter or something like that. And they then grow it in a solution of agar. And so basically I want to talk about next a quick rundown of that. So in 2011, most recently, they finally figured out how to grow it from seed, and that required actually dissolving the seed, stripping out the DNA, culturing that DNA with hormones in some sort of gel medium like this. And instead of it just growing itself like a grafted plant style tissue culture would, they actually had to encourage it and scoot it along with new chemicals and processes that they had found specific to that plant. So UC Davis, which is where I said I was streaming from today, I'm not, is the place that came up with that in 2011. That opens up a lot of new ground. So basically, the word clone, which is what a tissue culture is a Greek word, and it comes from slip or twig. And so slip as in a small slip or spot or piece, like you put on a slip, which is a little undergown for women, or clothing, small piece of clothing, or twig, which is a small piece of a tree. So basically it means a small little piece of something, and that goes into the solution, and it needs to be completely sterile because they found in the 1939, as I said, Haberland in Germany, the Austrian man in Germany, found out that unorganized cells can divide into things called callus. So that gives you a blob of a plant. So it's like someone took the plant and just like melted it into a living pile of cells. And so that is the first step when they're tissue culturing if you don't have the root system. is you basically got these little globs, and you can see right here where they started. So they inoculated this one in 6 spots, the center and around the edges. And same in this one, you can't tell because it's a little bit of a different style of plant. But in this one, you can also tell that they probably inoculated around the edges. As I said, you can actually see uh, the the tissue cultures coming apart right here because it's so hot today. I like the ADA and ADF when they were still running do this with clear versus black, because if it has really white fragile roots or green roots or something like that, they use a color that contrasts best. And also if the plant roots need to not be damaged by sunlight, then they will put it in the darker agar. And if it doesn't matter on that, or if the roots are pretty easy to see and easy to separate, then they'll use the clear. So the clear is kind of cool to see because you can see the whole structure of it. And when you're looking at a tissue culture at the store, what you're hoping to look for is you're hoping to look for clear delineation of different roots. You want to usually see, it depends on the plant, but you usually want to see some color in the roots, at least on the margin of where root and stem meet. And then you want to see that this upper area is nice and moist, because this is how this plant has been growing for anywhere from a couple weeks up to 6 months. So in the case of like Bacopa, it's probably been growing for only, a month like this. And so you had those calluses, which are those living balls of cells or piles of mushy cells. And they're all the different right cells that you need for a plant to form, but they don't have the information of how to organize without hormone cues that turn on that epigenetic code. Now, this wasn't known, but in 1954, a man named W.H. Muir, or Muir, made the first cloned anything in history, and he cloned a pea plant. And it was actually the same strain of peas that Gregory Mendel, who is the man who came up with. Can I get a shout out? Alonzo Zacharias. Hey, what's up, man? Check out his channel. Alonzo Zacharias. Thanks for coming in. I just said in this one, the first half is basically talking about just like if it were a video that isn't so interactive, but feel free to queue up your questions and I will get to them soon. We're almost done with all this history nonsense. But I'm explaining basically where these plants came from, how they're made, and the history that went into it, and the people that went into it, and why they're superior or not superior, and limitations and those sorts of things. So we've got the piles of green goo. It's protoplasm. Cell walls aren't forming right, and it looks like a plant that's melted. Oh, thank you, One Man Aquarius. He says, this is knowledge, brother, not nonsense. Thank you for that. Well, I appreciate that. That means a lot to me. I wanted to do this live stream as a video people could watch later and feel informed from the live stream, at least for part of it, because, live streams are really to interact with you guys. But this one, I have these plants. I had some money I spent. I got these at Aquariums In. And they are the shop, the owner, Steve, who has sponsored my setup here, which is kind of a mess at the moment. Actually, I'm redoing some things. But I got these new plants, and he gave me a hookup a little bit on some of the stuff and the tank setup. So aquariumsin.net. If you're in the Seattle area ever, check it out. It's all about aquascaping and nature, but he's got every kind of tissued plant that he can get his hands on. These are all imported. Now, I was saying what you're looking for when you're buying a plant. So the melt that you see in underwater plants, that is because of the epigenetic code. So the epigenetics of the plant say, oh my gosh, I'm underwater. I'm going through puberty, the equivalent of I'm going through a big life change. I need to switch the way I produce myself, and I need to switch up osmotic functions. That means how water enters and leaves cells. and as well as how nutrients are gathered and how sunlight is processed. And that can really change things. Like, you don't need a big wide leaf if you basically are trying not to get ripped off in the current underwater. You have a more narrow leaf. Or maybe you can't filter sunlight as well, so your leaves get big and bushy. So these plants are all going to be underwater plants, and they can live their entire life underwater, but they're started in, this would be considered immersed, quotes. When a plant is said to be immersed, it is started in a wet condition. This might as well be water as far as the plant can tell. So this is like hydroponics in a cup. That's what a tissue culture kind of is too. It's a cloned plant that's being hydroponically grown in a cup. So just the same as you would grow a tray full of clay substrate with nutrients injected into the water and have it flowing through, they came up with the proper chemicals, which I'm just about to tell you about, of how to grow that within a cup. And that's pretty incredible, especially for 1950s technology, 1960s technology. So yeah, let's keep going. So in 1954, W.H. Murr, pea plant, that was the same plant cloned as the guy who figured out, the monk Mendel, who came up with the Punnett Square, which allows you to see recessive and dominant versus recessive genes, and to see, like, in a pea plant, if it has blue flowers or pink flowers or yellow flowers, you can actually guess what flower color it will be, knowing which is recessive and which is dominant. And that is called a Punnett Square, Punnett, I believe. I don't know, someone can look it up if I'm wrong. So the Punnett Square, I've shown you that in other videos. You can look for that in my genetic videos talking about guppy traits and things like that. But, and shrimp as well. So that, in honor, was the plant that was cloned. So it kind of ties together into botany history, which is really interesting to me. So the next big thing that happened was they got that ball of goo, and the pea plant, out of the ball of goo, somehow he'd used root tissue, and that was the key. The root tissue, out of 1 in a million chance, started growing properly. And at that time, they were adding, I think they were adding, let me look at the exact list. Okay, so they were adding sucrose or sugar, and that was for carbon for them to grow as well as energy. And then agar or agar, which is for, they also use pectin or gelatin. So one's made from animal proteins and bone, or like cooking down, like in broth, you've got the thing you skim off, like in pho, that's a gelatin afterwards. So that's a gelatin, whereas an agar is more of a plant-based, like pectin type solution, if I have my things straight. And then they had myo-inositol, which myo-inositol is an osmoticum, and osmoticum means, so I was talking about osmosis, how plants change how they suck up and put out water, as well as things in the water, nutrients and things like that, to grow. Well, they figured out in 1964, these guys, Toshio Murashige and Folk K. Skug, figured out how to have a substance in the agar, and not just have a clump of stuff, but how to regulate the moisture. And so if it wasn't in a completely sterile sealed cup like these are, they rot. Other things take over that pile of goo that is an entire plant that has been melted. It's almost like if you put a plant in a vacuum, it's how it would end up, just like a pile of melted goo, which is what happens sometimes underwater to plants. The leaves turn into goo and fall apart, and then it regrows from the root ball or the rhizomes, and that's when you have the water form of the plant. So it's really similar in that way. So this team comes up with a solution, which is finally made public in 1968 after first done in 64. They've got pea plants, they've got other plants growing now as successful clone plants, And the problem is they were not growing very healthy. So then they come up to the idea of using a plant growth hormone. So they had to isolate the hormones in the plants, which had been going on since the 1930s. The Nazis were really big on this because they had been using hormone and steroid injections in people, such as anabolic steroids and things like that, to give people extra strength, as well as amphetamines and all sorts of things. I believe it was a Japanese chemist who came up with most of the forms of speed that are used today, as well as oxycodone. All sorts of medications were invented because of war, so you would keep going through pain and stuff. And they looked at plants, they looked at animals, and they tried to just copy what they saw. So they'd see like that plant is able to withstand frost. Maybe there's an enzyme in it that can make humans withstand frost. So there's all this research going on that was for, killing reasons, to kill people and make soldiers super soldiers and things like that. And the scientists often just wanted to do this research for the sake of finding out, for knowledge. But it wouldn't have happened if it weren't for some of the really terrible regimes, also our own regimes in the United States. And if you're a Japanese person who was living in Nagasaki, you might not think our goals were always so noble. So it's all relative, but bottom line is out of destruction came a lot of knowledge in First and Second World War, and that's when all these studies and sciences were first really figured out. Now, this is also the same time when we came up with chemicals like Agent Orange and DDT and all sorts of pesticides and things like that. So we had in the 50s and 40s, we were coming out with things that could manipulate plants and help crops grow, but they had side effects, and we didn't look into that. And so it took birds and eagles, like the bald eagle, almost went extinct because of DDT thinning their shells, and that was something that killed pesticides, or killed pests. Sorry, it was a pesticide. So now they're much more careful with how they do things, and it has to be done methodically, and in theory, it has to be done with a social conscience. So those two guys, Toshio Murashige and Folke K. Skoog, were working for Big Tobacco in the United States, and they wanted to figure out how to make tobacco plants. They made the perfect Marlboro cigarette, and they liked the way the tobacco burned, and it grew really fast. And this plant was just like, the Olympic medal winner of tobacco plants. And so it was the tobacco industry that funded 90% of all of this after the war. So the tobacco industry, and then ironically later, the marijuana industry, and a lot of horticultural work was also done with opium poppies, was done underground by underground scientists in many cases, and then passed on into legitimate places. So UC Davis is one of the places that dealt with cloning and tissue cultures the most. So they figured all that out, and they found out this combination of micro and macro nutrients, and it's a micture called Murashige and Skoog basil salt base. And that's macro micronutrients which trigger actual hormones and steroids in the plants. All of a sudden, they keep their form, and we have successful clone plants. Now, they're still in sterile conditions, and there's still a lot of issues with secondary contamination, and it will be a number of years before they understand that you need to use an autoclave, which takes all the pressure out of an area, and you sterilize the tools. So you may be using a piece of something, you may have something this size, and you may be using a piece, let me grab a piece of something here, this big, so compared to my finger, this big, and you put that in an empty cup of agar, and this is all sanitized, the cup is sanitized, and then there's a hood called a laminar flow hood, and it's the same for mushroom cloning, and if you're growing mushrooms indoors, You have to keep it completely sterile. There's all sorts of spores and tardigrades and little critters and things that float around the air all the time. Mold spores, things like that. And so you have to keep viruses, mold spores, all those things out of the way. And they keep a hood that sucks up air, kind of like the hood in your kitchen, and then gloves and everything. And they usually keep a burner, a Bunsen burner, that sterilizes the tools as you're working. And then they mist everything down with isopropyl alcohol, which you can do at home too. I'll probably do another video on that end of things later. But you'll know a whole lot about it, enough that you can probably do the research now and figure out how to start. So the last big things that got introduced are six benzy benzyl amono purine solution, and that inhibits or promotes, I should say, root growth. And so that allows the structure. It's a hormone in plants. It allows the root and the plant stem to be two different types of tissue. Not all plants are like that. In aquatic plants, like I'm trying to think of one, Worcester Tongue, it is actually a type of gamophyte. A gamophyte is, like I was saying, before those epigenetic tags get turned on, they are prepubescent, to draw a bad comparison, but it's as close as I can think of explaining. And so Siswassertang is this clump of gelatiny looking plant seaweed stuff. And if it's out of water, it's like a thick ball, and it turns into, let's show you over here. I know you guys have been looking at a towel too long. So Siswassertang, I'm sure there's some in here somewhere, is actually, they thought it was a fern. What is it? Blah, blah, blah. But it starts out as a slimy group of cells, and then it will, as I said, once it's out of the water, or it can go the other way too, turn into little ferns like the ones back in here, and then it'll grow outside the water. And this plant right now looks so run down. There's algae on it too, but it's because it's transforming into its water form. I think I have a better example over here. So these are the last ingenious inventions in the tissue culture sciences. Is basically they figure out all these hormones, and they don't have to just put plants in a good condition. There's some right there. So that's Siswassertang that was from land and is still turning into. So the Siswassertang back here, where are we? Let me get my finger. It's going to be huge. Swassertang back here, that green piece there, and back in here. That's what that looks like. There's a video on Siswassertang. It's really interesting. Siswassertang does not age. It is an ageless plant, so cellularly you can clone it indefinitely. It clones itself already. And if they had started tissue cultures with that, they would have probably thought that it was a piece of cake, just because of the nature of all of that. So yeah. Now, before that, though, it looks like this. It looks like a Pelia or some sort of, gamophyte is what they call it. And it looks more like a thick moss or something. It looks like, I'm spacing on the name of the organism name we have for that plant. But in any case, that's kind of explaining like the form that those hormones can cause the difference in. And so the last thing that they came up with is naphthyl inecetate acid, and that is a way to, in this sealed container, put months worth of oxygen fixed into the agar. And that allows it to have the oxygen. Then they seal this up usually with CO2 and nitrogen gas, as well as some oxygen, to represent the ideal mix for plants to breathe. And then the water in here is drawn from the agar, which used to probably be up here, and then it condenses and condenses. So that is pretty much the history. That is the long history of how we've come to get tissue cultures. And the last step in that, in the 80s, we got potatoes, all sorts of, bananas, apples, fruits, dates, strawberries, raspberries, all sorts of things now are grown this way because you can now just take, if you've got it all clean in an industrial setting, you can take pipettes and beakers, just like your water parameter testing API test beakers, and you can inject 10 at a time. And with the air being sucked out and it all being sterile, then they come back a week later after they put it under some grow lights, and ta-da, they've got a start of, say, a rose. And it looks exactly like the rose that won the World Rose Championship earlier in the year or whatever. So it's just the same as if you had a really beautiful cichlid or shrimp, and if you could clone it, like for plants, why not do that? Same with nutrients. If they get a weird freak of nature plant that seems to be surviving, but it produces 10 times the amount of fruit, that has 10 times the amount of vitamins, or if they engineer that and it's not stable most of the time, but one time it works, they can do that. And now they're starting to do that for aquascaping. It wasn't until 1992 in Japan when they finally came out with several hormone cocktails and were able to figure out how to do um, basically hardwoods. So things like pine trees and bark, they hadn't been able to use that, just like seeds were giving them trouble because it was a dead cell. It was not an active cell. It's the outside of the tree and it's the heartwood of the tree, or the ring around that that is alive in trees. And so they were having a hard time without that live pulp cells to do that. Now, once I've planted this, you can see all these tiny little starts are starting to take over and run. So let's go one more time over to this aquarium, then it'll be question time. And I know this ran a little long, but it's a detailed history, and there's a lot of little side things that I wanted to inform you of along the way. So thank you for bearing with me. But just like in this tank, all of these plants right in this immediate area started in that other high-tech tank with the CO2, grew too fast, got covered in algae, and then in here, all my algae eaters decided to clear it out. Harrison Lucas, you said, shoot, I seem to have missed the history part. It's going to be up online in live stream form, and that's exactly why I did it first. So people coming to see that can come to see that. Everyone else, now is your time. You can ask questions. If you're done with the history part and you don't want to hear any more, you can tune out now. But if you stuck around for 45 minutes, please like, subscribe. Feel free to share. I mean, this is a hobby that very, very few people and no companies in the US are doing in-house large scale, other than universities and some classrooms and things like that, and a few people who are into it, surprisingly for marijuana, mostly for strains of marijuana. In my state, Washington, we're in Colorado also. We have been at the leading edge of this now for over a decade of plant hormones and things like that. And so you will get these little plants. Those cultures are such a good deal because they're not just one plant that's already stuck in its ways of it's going to grow spindly here at the base and you have to retrain the tops, yada yada. It can be anything, and you've just got a little piece of it and it's ready to be molded like putty for you. And so you can put it in a gravel tank with root tabs, which are basically the equivalent of that same auger it was just in, but it has the oxygen flow of that. Or you can put it in a tank like this where it's getting CO2 and it's getting live substrate that is pumping in all sorts of nutrients and things. But yeah, so if you like this, if this information is good to you, there's a whole playlist about plants and history. There's a whole just history talking about plants and fish. And then there's also a Facebook group for help and things if you're not getting an answer that you need on Facebook or on YouTube. Sorry about that. But another cool thing about this is like Nana Petite and Bucephalandra or Bucephalandia is that they can grow it quicker by and by the way, this is going to flower soon, this boost that I have in here. It's crazy how quick things grow with CO2. But yeah, it's really beautiful when you have tissue cultures of certain things because they grow in, you can break it off wherever because they're basically all stem cells until it's gone through that epigenetic change of thinking that it's an adult plant. So you can literally take a scalpel and cut this crypt right, like even though it looks like there's 6 plants, and when I open these, you've seen in other videos when I've put them in water, soaked them, taken off this gel, and then carefully kind of loosened the roots and just cut where there's the best root. Um, from above, this is a rare ultra pink flamingo, as I had said earlier. And instead of having to give one little trimming from Steve, who owns Aquariums In, he can put it into a container like this and make he can make a tissue culture clone of it. Now, some of these get really expensive. This one, as you can see, was $18.99. And usually what that's reflective of one is the journey that it takes to get the plant, but two is also, well, demand. But it's coming over from Japan, so you figure then they have to, the retailer has to have a 50% markup usually. So it was probably $9 coming in from Japan or something like that. And they had to grow it for probably 5 months. So with Bucephalandra, things like that, the slower growing plants, even though they're growing much faster because they're at the ultimate speed they could grow at, because they've fine-tuned these things, every plant they have to figure that out for. And some are much better at it, and they have a terminal life. So this will only grow up to about this high, then it'll start to bend over. And so this needs to come out of here quickly. Yesterday, this was not this tall. So it's hard for plant retailers to keep these on hand, even though they're a little easier than live plants sometimes. They cost more. But in this right here, right now, if I wanted to, especially in a tank with CO2 and fertilizer, I could cut all of these in half and then use the root system, which were all stem cell roots, and I could cut up that root system. It would grow into the substrate, eventually come up, and then the photosynthetic tops would grow. I probably get 20 starts for $11 of this rare Bacopa. So it can be a really, really economically effective way to buy plants if you're patient. This entire back row in this tank was done with cultures. They all started, this small, and I cut the top off and replant it. You can see where I cut the last top off because there's algae left. So that was where I cut it before, and then I put it in the other tank. Here's those erythromicrons I was talking about. They're my little buddies. And yes, I know people are going to lecture me that I have erythromicron or emerald dwarf rasboras with celestial pearls. I know they are able to hybridize, but don't worry about it. I'm not going to put them back in the wild. This tank also almost completely all done with everything in it other than the pogostem and octopus, this big guy here. And let's see what else wasn't done that way. One of the sword ferns wasn't. Everything else, these crypts, these were little. So all these crypt undulatas here, 1, 2, 3, 4, 5, they all came from one cup. So they all came from a cup. You can surprisingly find these things at PetSmart, Petco, things like that. The selection's not great, but also H2O Plants has some of that stuff. Aquatic Arts might have some of that stuff. And if you go to Aquatic Arts, you can get 10% off using the code SECRETSHRIMP10 in all caps, no spaces. Here you can see some boosts that I am using. Now this is low tech, so this is not just for high-tech tanks. This tank is loaded to the brim with fish, like way too many fish by any standards of other people. I have a power head in here, so they get a treadmill essentially. And they have lots of hiding spots down here and substrate for plants and a maze of plants. All of these ones in the front here were also tissue cultures. The Java fern, stuff like that, not a tissue culture. The lily, not a tissue culture, but I have cultured 2 new starts of that lily. which is Nymphea micrantha from Egypt and Sudan. Also, the guppy grass is not a tissue culture either. Don't bother with super cheap plants that grow like, weeds, like Java ferns. Don't bother because they'll turn into these and then you can just plant those. Like they've hung onto my filter sponge. Like I can't get them to stop. But all these plants I just replanted last week, I could cut them all and I'd have double the plants because those are now old enough for one, but two, they are tissue cultured plants. So really cool plants. I wanted to share all that info with you guys. We're going to take a peek because somebody asked if we could take a peek at everybody's favorite, the shrimp downstairs. But now is a great time. Please ask any questions. Also, I'll say that I have been using Easy Green. Okay, so let's see here. Let me read the questions. I got some 1-2 grow exclamation point Pogostemon and Elocarius Acicularis mini. Cool. Let's see. Shoot, I seem to admit. Yeah, I figured out. Whoa. Okay, I just watched the whole story. I'm just wondering, when do you breathe? Damn, good job. Tanks are looking good. Nice plants. Thanks, man. I don't breathe. I'm actually a tissue cultured human. I am the $6 million man, or at least my insurance company has been billed as such. So, yeah, last bit of the video. Feel free to ask any questions you have about tissue cultured plants. They're really popular in Europe and Asia. And also they're great because they're not going to bring in any invasive species. And these tanks have a couple tissue cultured plants, but these are basically not tissue cultured plants. And you'll be able to see the growth differences. So if the light didn't crap out on me, sorry about that, guys. Let's see here. So they're more spindly. They're not as dense. So this is a crypt that is just kind of growing a little crazy. This one's still doing okay. These are LowTech tanks too. This tank in particular though has, you can see a green dragon plico. Let's see if we can show him. And he's a good 5 or 6 inches long now, and so he is able to rip everything out of its place, as well as the Bapindi cribs that are in here and the Cory catfish. They rip everything out. Here's my new green shrimp. Aren't they pretty? Oh, if they'd focus. Down here, again, water sprite, water wisteria. This is basically also what you could see without cultured plants. Totally normal, healthy growth, but you also see the tanks covered in snails. The shrimp are in here. These are my cold shrimp. I wish I could clone my shrimp because I got some that I really like. I found out a little bummer this morning, and I haven't pulled them out yet. Not really this morning, but a couple hours ago. One of my black crystals, or black pandas, passed away, so that's a bummer. But my black King Kong and my black panda are doing well. My other black panda back there, so they're all doing well. It's a really hot day, and here's again another crypt that wasn't a tissue culture. You see how loose it is? It's not reaching up quite as much. It's just kind of holding on. And then also in here, if you can see it, we've got baby shrimp, Neocaridina from Lucas Brett's. We've got little teeny yellow sakuras and little teeny fire reds that were the world champion at the Chicago Shrimp, the Aquatic Experience last year. And I also have I have his blue dreams also. I found out an interesting fact about the blue dreams in that in this room now where I'm getting sunlight, basically the blue on these guys turns dark blue when the sun's on them. So it's a form of active camouflage, whereas they're kind of a brighter blue And at night, they're pretty translucent. So you'll notice that with cherry shrimp too. You can grade them a lot easier and see which ones have red flesh like Bloody Marys versus which ones don't. But these tanks are supposed to say 72 degrees, and they are, because of the temperature in the room, they're inching up to almost 75 right now. Looks like the shrimp are doing okay, but it's the caridina that I fret about. And there's also gold bees in here that are pink. I don't know if you can see that they're pink. And then there's crystal reds. And then I've also got, and I'm hoping with the warmer water that I don't get the green rot on the eggs, but I've also got three now, I believe, buried blue, the world's greatest blue strain, as Lucas has titled them, because they breed very, very true. Okay, so questions. Hello, love the channel. My amonos are pretty, let's see here, are pretty aggressive with my blue dreams during feeding. I have 3 amonos and 10 blue dreams in a 5-gallon. Should I rehouse the amonos? I would. Amonos get pretty big, and they'll actually, as far as I've been told, I haven't witnessed it firsthand, but I have now been told by 4 people who I really respect that they will for sure kill baby shrimp and eat them, and actually will fight slash kill somehow. I don't know what they do. They poop on them to death or something. I don't know what a shrimp does for defense. They don't have claws, but apparently they can bug shrimp, stress them out. For sure, you can stress shrimp to death. I mean, even banging on the glass too much can cause a shrimp to die, especially like Taiwan bees and things like that. Now, blue dreams are more stable, kind of like cherries, but I would not. If they're picking on them every time you feed, you may try using feeding dishes like this and putting out like 3 and see if that breaks it up. But honestly, if you're trying to breed them or anything, I would separate them. The blue dream shrimp are not really helping eat a lot of algae. By the way, here's more Worcester Tongue in its aquatic form. And actually, you can see it in its non-aquatic form unraveling and turning into it. So that was brought from upstairs. Sorry, off topic, but I was wondering, you're on the West Coast. Do you have earthquake-proof tanks? No, I do not. I wish I did, but no, we don't. If there's a little earthquake, it'll be okay. Um, we'll get some sloshing of water. Yeah, so here's a buried blue. Here's another buried blue, and then there's another buried blue. So now I have 3. Right now, though, you're speaking of fighting. This is like fighting for them. And that can cause a female to lose her clutch of eggs really easily. Even moving their tanks, like I've got a green jade in here because you'll have to watch my old videos. I highly recommend anyone watching to please, please go back and look at my old videos that are from, I'm not an old channel, but January, or go through the playlists and look at stuff that doesn't get suggested because I had like 50 followers or something, and just compared to now, and when I say Taiwan bee shrimp, everyone goes crazy and clicks on it, 15,000 views later on that video where I spent hours on the history of aquariums, or what the L numbers mean in pleco, things like that. Yeah, so, all right, let me see another question. You can small fans blowing over the top of the water to keep temps down. I just went a full week without AC in Phoenix this last week, and I was able to keep all the tanks below 75. Yeah, uh, you can. You can do that. You'll lose a lot of water through evaporation. And by the way, I actually do have an AC for when it gets desperate. And then I have, compressed air and CO2 canisters. And a fan. 2 fans. We got a box fan upstairs too. And so that's exactly what we've been doing. But there's a cross breeze through this window, through the window on the other side of the house, even though it doesn't feel like it. But this water was changed out 2 days ago to the top, and it's already that low because of how hot it's been the last 2 days here. But pretty much no one has AC in Seattle, and we have an AC unit that's old, and it was our roommate that used to live here, left it here, and basically it is enough for maybe a 10 by 10 bedroom, and that's it. So, but yeah, so I think I'm getting near the end of rambling on, ramble on. So I'm gonna lay down. I think I am getting uh to be a sweaty yeti. Um, yeah, so it's hot in here. Yeah, for Seattleites, we're used to like 60 being a warm day and overcast. What's the CO2 pressure should be? What's a CO2 pressure should be on CO2 atomizer? Okay, so I'm gonna try to interpret that question a little bit differently. So you said what pressure should the CO2 atomizer be at? So this is it's an atomizer is something, or diffuser is something that actually puts it, it's usually an inline device, so it runs to a sump or a pump, and it has a water line, and it actually swirls the CO2 into that to dissolve it, whereas this, the CO2 clings to things and goes to the top. But this is my pressure gauge setting for my CO2 on a 2.5 pound, but on the 20 pounder downstairs, that giant one, same deal. So the can itself going into the regulator is about, I don't know, 950 psi, and the smaller unit, which is going into the bubble counter, the low pressure is letting it out through the needle valve at 55, 56 psi, 54 psi. Anywhere between 40 and 70 may work, as long as you use CO2 line, because I've used non-CO2 line and had it blow up before. So don't do that. And when you turn it on, ease it on. So just really barely, like here, it's like barely a turn is the difference between this, and not even a turn is this. So, and the other thing is, get an airlock, water lock, gas lock. These are 50 cents or something. Usually you can get them at Petco or at a local fish shop. But yeah, they allow water not to go back up in and screw up this chamber. And then this is the bubble counter, and I usually allow it on this tank because I have a hang off the back filter and a power head in this that is causing surface agitation. There's a good amount of oxygen, so the more oxygen you have, the more CO2 you can pump in, but you can see all the bubbles coming from the hang off the back filter right now, and I need to clean this tank up. As I was saying, I'm redoing it. I used to have this arch in the tank, and with the amount of algae growing, it's just too hard to keep up with cleaning and not collapsing it on the shrimp colonies that are forming underneath all that. So I need a different setup where I need to mount it all together. So yeah, but that's what's going on. And then I like aquarium co-op stuff because it's easy. But essentially this stuff that's dried around the edges, that is all of these macros. So nitrogen, potassium, phosphate, magnesium. The trace elements are like manganese, copper, zinc, stuff that can kill plants and fish normally, but in really, really small amounts, it's okay. What fish do you have in your shrimp tanks? That is a trick question. I have shrimp in this tank. I have amono and caridina in here. 5 amonos that are very young right now, and I'll have to move them when they're not, to help while this tank has cycled. It's 6 weeks old now, so it's still got a lot of algae and stuff forming. I'm fighting it with plants. hence the floating plants and me just throwing random plants in as much as possible. I took out all these plants, and as I've said earlier in the video, replace them. Hold on, let me turn off this thing on the floor that is still running. I didn't realize the all here, and they're growing tall. But I use also flourish iron, and you can use Excel or carbon and stuff, but with the CO2 I don't use any of that. I take the carbon filters out of my hang off the backs. But I've got cherry shrimp in here and Malawi shrimp. I've got uh, I've got all sorts of stuff in here. Yellow sakura shrimp, but we've got plecos, ottos, endlers, rasboras, danios, tetras, and then in this tank, just tune into other videos, but this was my most successful shrimp colony until recently. I put some adult cribensis poultures in here, and they hunted down the shrimp in less than 48 hours, all of them, like 100 and some of them, at least. But everything you're seeing here got along with the adult shrimp. Now, the baby shrimp, look at my old videos about how to make a cave for your shrimp and how to save their life. And there's lots of good material online by me, by LR Bretts, lots of other people too, on how to protect shrimp. So they basically need a series of chambers, like these areas where the fish can't get into, if you can see that through the reflection, that's too narrow for the fish, but that the shrimp can hide in, and the babies will hang out in there, as will the mothers. So, yeah. Thank you so much for subscribing. Thank you so much for watching. I'm just reading some questions. If you're still having problems with your CO2, message me or join the Facebook group by the same name, and I'm sure somebody in there will help. Also on Facebook, the Planted Tank forums and the like high-tech tank forums, they'll all let you know what to do. There's a lot that you might need to provide information-wise, like the model and, you know, that kind of stuff. Of that and how big your tank is and what your temperature is and what you got things set at, yada, 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 yada. So, but yeah, and you can see that this stuff's starting to break down and stratify. So the plecos are in here and they're growing super quick because they can with all that algae. But thanks for watching, and if you stuck it out this long, I highly appreciate it if you subscribe, hit that like button, and if you want, share it with this tissue culture thing, or if you didn't see it all, go back to the beginning and learn some history in a little bit of a rambling way, but I like to tell it like drunk history, so to speak. I'm not drunk. I was just drinking Dr. Pepper and I'm hot. But, oh, Damian Bloodstone, how's it going, brother? Take it easy. I hope you're doing well, staying cool. Sure hot here. You're probably used to it. But as I'm sweating and want to go rinse off, I'm going to leave it to y'all. Try some tissue culture plants if you haven't. If you go to aquatics..com, sometimes they have tissue culture plants that they have grown out, so they'll get them as tissue cultures, like rare stuff, and then they'll grow it out and sell it. Like their amazing red temple plant. And go there, use the code SECRETSHRIMP10 at checkout. You'll get 10% off. The shrimp that I just showed you, they are, a good deal of them are from Flip Aquatics. If you go there, flipaquatics.com, for like Caridina shrimp, really, really nice imported shrimp that are top class Taiwanese if you want them, or Japanese or Singaporean. Use the code history with a capital H and then 10, the number 10. So history 10 and all one word, no spaces, and that will get you 10% off your order from Flip. Both places, aquaticarts.com or Flip Aquatics. Please just say, hey, I got it from Alex Williamson or The Secret History. And the thing is, both places have made a deal. I said, I'm not going to feature your products as much as like I'm stoked to get, pay 50% as much than normal on products. I'm not going to feature them unless the people viewing get a deal too. Like they need to get a deal. If I'm getting a deal, they need to get a deal. And you're getting a deal as those companies because you now will get more business. And they're like, totally, totally. They're Rob, the owner of Flip Aquatics, totally cool about it. Preston, and Dave, the guys in Point Contact, are really great at aquatic arts. And yeah, so also H2O plants for if you can't find tissue cultures other places, order from them. Let him know, Justin, who runs H2Oplants.com, let him know that you found them through this channel and ask him about any specials. At one point, there was a special for people on the channel, so he might hook you up with a little free something or discount or something like that. So, but I don't know for sure on that one anymore. But I want to help you guys out if you're interested in plants and stuff. This is like not any shameless plugging. The channel's not monetized, but I do like doing unboxings, getting stuff that I can then talk about, like those cultured plants from Aquariums In. And instead of paying $20, I pay $10 for one of the plants, or one's free when I buy four. I get some sort of deal usually. I'm going to be transparent about that. So yeah, but I hope you guys all learned something. If you want to support the channel growing and me getting better filming equipment, being able to set up the chat a little differently, some software where I can do interviews live with people like L.R. Brett's buddy, who I'm doing art for, as well as like these endlers came from his strain, but I've turned them into leopard endlers with blue tails and black up by their head. So, but yeah, to all of that, Patreon, if you save some money, if you go to Aquatic Arts or you go to Flip Aquatics and you save, you get the free shipping because you ordered enough, and maybe they throw in an extra shrimp when you order 10 or something, plus the 10% off. If you want to kick out, you just save $10. If you want to just for a month or something, kick me $1 on Patreon. That's a great way to support the channel too. Liking and sharing is another great way. So that's enough, whatever, talking about myself. It used to be really awkward to me to do that. What's up, Mick, too? Sorry, I didn't see your text till just now. So, but yeah, I'm about to sign off. I'm losing it it's 92 degrees up here according to the fish tank's external thermometer. So it's real hot. I live in a metal roof house for some who knows why reason on the 3rd floor. So it's real hot, and this deck is stucco-y, and it just reflects white light, and then there's metal and a flat roof with metal siding and no insulation. So it gets literally up to 20 degrees Fahrenheit hotter inside the house than outside. So I need to go lay downstairs with my lovely wife and melt into the bed like a plant that has not adjusted to water. So thank you so much for your time, you guys. Take care of yourselves, take care of your plants, and I will see you next time. Swim on, guys. Goodbye.